So the story that I have to tell starts about 3 years ago. I had a meeting in my office, not this office. I had another office then with Doug Melton. Told him about a science project that we were doing in the lab, and he just thought it was really cool and exciting. I was actually telling him because I thought it was really cool and exciting, and he recognized that this was potentially going to be transformative. So he said, I think the HSCI should support this idea because it's really key. And that's actually not the reason I was having the meeting with Doug. It was just, happened to be talking about science. But he immediately sort of cobbled together that this would be something that we wouldn't be able to get funded by any other way. And if it was going to happen, it needed support. So Doug basically suggested that I make an application, which I did for the HSCI, and we were granted seed funding. And we had this idea for making iPS cells, but doing it in a non-integrative manner using RNA. RNA is a non-integrating vector. But as it turns out, RNA just wouldn't work because cells recognize RNA as basically a virus, and they respond with a terrible antiviral response. So we modified, through our research, we found out that if we modified the RNA in certain ways, we could basically make it stealth RNA, and it wouldn't be recognized by these antiviral pathways. It would express proteins, and we could basically affect reprogramming. So that worked after about 1.5 years, and that was funded the whole way through HSCI. But it had actually more utility than that, because basically what we developed was a kind of a platform for expressing proteins in cells using this modified RNA technology that we developed. So in addition to reprogramming cells to pluripotency, in the same paper that we published, we also used that same technology to now take the fate of those iPS cells and turn it into a terminally differentiated cell type. In this case, it was a muscle cell. So that was pretty cool. Now we could both reprogram and direct the fate of pluripotent cells using the same non-integrative technology, which, as it turns out, did both of these tasks with very high efficiency, another bonus. But it became evident to me that we could actually synthesize any protein in a cell using our technology. And as almost everybody knows, there are many patients walking around the planet that are in desperate need of many different types of proteins. So I thought, well, we could apply this technology to possibly meet unmet clinical needs. So about exactly know the time, but something like 2 years ago, or 1.75 years ago, something like that, I decided to start a small company, which I did through some venture banking in Cambridge and with some co-founders, including Bob Langer at MIT. And we made a company called Moderna. As it turns out, that small startup company has turned out, it's really done very, very well. So it now employs 25 full-time employees and maybe 10 sort of part-time employees. So it's employing a lot of people. And very excitingly, I think it has a real potential to impact patients. So a clinical trial is planned for next year, so it'll be in patients next year. We've put this platform into mice and rats and monkeys, and it works very, very well. The fact is there are, as I said, many different clinical settings where proteins are needed. Almost all genetic deficiencies are deficient in a protein, so you can imagine applying this technology to that. So I think it has a really an enormous potential to impact a lot of patients' lives. And basically the story starts with a meeting in my other office 2.5, 3 years ago. HSCI was just incredibly supportive of the whole thing, which as I say is 10 months away from being in patients.